Well, good afternoon. If you're on the East Coast, I think we have a couple of West Coast folks. Good morning. It's my pleasure today to introduce Nathan Sedghi, who's a PhD student, and he's finishing up after 4 years of hard work trying to chase nitrogen around agroecosystems. And he's got quite a bit of interesting data to share with us today on how we can improve the way we do cover cropping here in Maryland and in this kind of climate. To make it much more effective in capturing nitrogen, keeping that nitrogen out of the Chesapeake Bay, and hopefully keeping it where it's valuable on the topsoil of the farmland. Nathan, let me pass the floor over to you, and I'll be all ears for your presentation.
Thank you so much, Ray, and thanks everyone for tuning in today. I'm happy to tell you about the work that we've done over the last few years. Cover crops are crops which are grown in between the cash crop growing season. In Maryland, cover crops are very common. In fact, according to a SARE 2017 report, Maryland has the highest percentage of cover crop acres out of any states, and most farmed acres in Maryland use cover crops. Cover crops do well in Maryland because with our mild winters and year-round rainfall, there's potential for good growth. Mixed stands of cover crops are still rare, but they're becoming more popular. Currently, the typical cover cropping practice in Maryland is to drill a single species after cash crop harvest in the fall, and then kill it early in the spring, several weeks before the cash crop planting. Now, drill seeding is a practice where a farmer cuts open the surface soil, drops the seeds in so it has good seed to soil contact. It can lead to uniform stands, but since this has to occur after the cash crop has been harvested in the fall, there's a limited growing window. Cover crops are popular in Maryland because we have a program which pays farmers an incentive of $100 to $200 per hectare of cover crops that they plant. And this was start of an initiative with the Chesapeake Bay program for nitrogen pollution prevention. According to the MAX 2020 annual report, in Maryland, we had about 198,000 hectares of cover crops planted, and they estimated that this prevented about 1.6 million kilograms of nitrogen from entering the bay, and that works out to about 8 kilograms of nitrogen per hectare of cover crops planted. The base payment for the cover crop program allows cereal rye to be planted by November 5th. If a mixed stand is being used, which qualifies for the program, and it has a legume or a brassica, then it has to be planted by October 1st. But the program also has incentives for farmers to plant earlier. So this image is taken from Staver and Brinsfield. They did 5 years of work in Maryland, and even though there was slightly different weather each year, the pattern held constant that the water table recharge season in Maryland occurs in between November and about April is when it peaks, and that's when the water table starts drying out due to high evapotranspiration. So that's when the leaching season occurs. As the water table's rising, that's the rainwater washing through the soil. That's when the nitrate can leach through the soil profile from November through May. There have been several meta-analyses that have established the importance of cover crops in reducing nitrate leaching. A recent one by Thapa et al. Demonstrated within the literature that cover crops were more effective on coarse soils than on finer soils. They're more effective if there was a large spring cover crop biomass, and more effective in drier years than they are in wet years. Recent work in this lab by Hirsch and Weil is documented the amount of residual nitrogen within a soil profile. So these samples were taken across 29 fields in the mid-Atlantic region on commercial farms. And these samples are from a time when the cash crop, either corn or soybeans, has stopped taking up nitrogen. And they found that there was about 115 kilograms of nitrate nitrogen left in the soil profile. And a little over half of this is beneath 90 centimeters down to 200 centimeters. And that's important because this deep soil nitrate is very susceptible to leaching at the beginning of the recharge season. Nitrogen is difficult to manage in agriculture because while it's an essential nutrient, it's also incredibly mobile in the environment. There are multiple nitrogen species and chemical transformations that make in the soil. Nitrate is an important water pollutant because it's very soluble in water and it can leach away readily from farm fields where it's applied. This is a concern in the Chesapeake Bay because there is a history of hypoxia and fish kills and seagrass degradation. The major pollutants of concern in the Chesapeake Bay are nitrate, sediment, and phosphate. And farmers participate in a nutrient management program in Maryland aimed at helping to reduce nitrogen and phosphorus loading. And cover crops have the potential to reduce losses of all three, sediment, nitrogen, and possibly phosphorus as well. Nitrous oxide is another major pollutant of concern because it's a very powerful greenhouse gas, roughly 300 times as strong as CO2 with its heat trapping potential. And in ag soils, it's most commonly formed via denitrification using nitrate as a substrate. And the peak emissions of N2O from ag fields typically occur following nitrogen fertilizer applications. The conditions that are necessary for denitrification to N2O are when there's nitrate present, there's a soluble carbon source for the microbes to use as energy, there's sufficient heat so that the microbes can be active, and there's low enough oxygen conditions so that nitrate's a terminal electron acceptor, and that occurs when fields are really wet. Looking at the literature, there's some studies that show increased nitrous oxide emissions from using cover crops, some studies that show cover crops decrease nitrous oxide emissions. So really, there's mixed effects, and it's possible that cover crops could reduce N2O emissions by reducing the residual amount of nitrate that's used in this reaction. It's also possible that cover crops could increase it by adding a soluble carbon source to the soils. The mechanism by which cover crops could reduce the amount of nitrate leached is one by decreasing the total drainage volume of water through the soil profile. They do this by taking up soil water, increasing evapotranspiration, and that decreases the amount of water that can wash through a soil profile. They can also lower the concentration of nitrogen in this soil water. So as soil organic matter at the surface soil starts decomposing, nitrate's produced during that process, and the rainwater can pick up this nitrate and leach it away, but cover crops can take up the surface nitrates before it leaches away. And another mechanism is that cover crops could actually clean out the profile of this nitrates before the leaching season really begins. So in order to do that, they have to have deep roots that can access that deep pool of soil nitrate that discussed earlier. And in doing so, they'll clean this nitrate out, and the water that washes through the soil will have low nitrate concentrations. So some important research questions that we aim to answer. Is a radish clover mix with rye as effective as rye or radish alone at reducing nitrate leaching? Rye and radish are both well known to be nitrate scavengers, and it's possible that by adding a legume to the mix, the potential to remove nitrate from the profile could be reduced because a legume fixes nitrogen. So we wanted to test to see if the mix was just as effective as these cover crops that are well known to be effective at reducing nitrate leaching. We also wanted to test if planting a cover crop earlier into a standing cash crop will reduce nitrate leaching over the winter, and if interseeding can increase the cover crop biomass. So that's comparing interseeding a cover crop into a standing cash crop versus the practice of drilling after harvest. And is interseeding a cover crop a practical option on commercial farms? We also wanted to know if a radish, clover, and rye mix will increase nitrous oxide emissions over the winter. Radish typically winter kills when temperatures get really cold. When that happens, the radish dumps nitrate out at the soil surface, and that could potentially be a substrate for increased N2O emissions. So we wanted to see if this mix increased N2O emissions over the winter. And if extending a cover crop growing season, both by planting a mix earlier in the fall and killing it later in the spring, would affect the total nitrous oxide emissions throughout the year. Hypotheses we've tested. The radish, rye, crimson clover mix will reduce nitrate concentrations in the leachate as effectively as either the radish or the rye planted alone. The amount of nitrogen leached during the winter to spring period will be inversely related to the cover crop biomass and nitrogen uptake in the fall. Interseeding cover crops early will produce greater winter biomass than drilling after harvest. Winter nitrous oxide emissions will be positively related to the radish biomass at the time of frost kill. And cumulative nitrous oxide emissions during the cover crop growing season will be lower than the N2O emissions following the nitrogen fertilizer peak. So we tested these hypotheses with 4 field experiments over 4 years. For experiment A, we wanted to test the single species versus the mix. So that was rye alone, radish, radish, and crimson clover versus a no cover treatment. For experiment B, we wanted to see how the timing of planting this mix affected their ability to reduce nitrate leaching. So that consisted of 4 different drill dates into an open field in the fall. And these dates were separated by about 2 weeks on average. We compared that to a no cover control. For experiment C, we wanted to test how effective interseeding into standing soybeans was. So we had 2 early interseeding dates compared to drilling the same mix after soybean harvest with a no cover control treatment. And finally, in experiment D, we wanted to scale up and try this work on commercial farms on the Eastern Shore. So we worked with several farmers over 3 years, and on their fields, we had them early interseed a cover crop mix and drill the same mix after harvest and compare that with no cover control. The first year we did this experiment in 2017 to 2018, we didn't have a no cover strip in the middle. We added that for the final 2 years. Cover crop biomass was measured by sampling the cover crops at random locations within each plot. So we harvest all the cover crops that were growing within a 1/4 meter square quadrat. And these were separated by species into labeled paper bags. They were taken back to the lab, oven dried and weighed, and then they were ground up and homogenized, and we submitted them for LECO carbon to nitrogen tissue analyses. Cover crop biomass was measured in the winter, right before a time that we expected the radishes would winter kill. And in the spring, cover crops were sampled before any planned herbicide applications. In order to determine how effective planting earlier was, we calculated cumulative growing degree days. We didn't just use the planting date to determine the effect of biomass, because a day in September is much more important for plant growth than a day in November. So cumulative growing degree days was calculated using the formula, the daily mean air temperature minus 5 degrees Celsius as a base, and we added that up in between the date the cover crops were planted, and we used a cutoff of an expected dormancy of December 1st. So we added all that up, and that was the cumulative growing degree days available for that cover crop planting treatment. Nitrous oxide was measured only on experiments A and C, and that was done by installing rectangular metal gas chambers. They were hammered into the soil about 5 centimeters deep. And we installed these at locations within each plot that appeared to be representative of the overall plot treatments, while ensuring that each one of the chambers included at least some of the legume and the grass and the radish where that was the treatment applied. Sampling was done by closing the lid. There was a thermometer inside so we could measure the temperature inside the chamber, and we had a stopwatch. So once the lid was closed, we took a sample at time 0, and there were 4 samples that were taken in 7-minute intervals. And then we took them back to the lab, analyzed them on a GC for nitrous oxide concentrations, and then used the dimensions of the chambers and the ideal gas law to turn that into the amount of nitrous oxide produced. And by having those 4 samples taken, we could get the nitrous oxide emissions over time. And this was performed in between December was our first sample, and we ended at the middle of July. And we targeted our sampling at events where there was expected high peaks, such as when the radishes winter killed, we sampled for several days after that. When the soils would freeze and then thaw out over the winter, we took samples. Over the summer, we followed rain events, and also we followed nitrogen fertilization over the summer. And we continued sampling over the summer until the fertilizer peak had ended. We couldn't detect that signal anymore. Because we're sampling when we expected maximum values, we performed an exponential decay function down to a baseline from that peak where it was sampled, and then added up the area under the curve to get cumulative nitrous oxide emissions over the season. Nitrate leaching was measured by installing tension lysimeters to a depth of 1 meter. So this just shows what these lysimeters look like. These are made out of PVC or plastic, and there's a ceramic clay tip that's put in contact deep into the soil. And the samples are taken by applying a hand vacuum to the lysimeter. And that's done for about, the vacuum's left on for about 24 hours so that the negative pressure can pull soil pore water into the lysimeter, sample builds up, and then we come back and take the sample into an apparatus and a labeled clean centrifuge tube. And these are collected during the cover crop growing season about every 2 weeks. They're transported on ice back to the lab and analyzed on a latchet flow injection analyzer using the cadmium reduction method to get nitrate concentrations. Now, while doing this, there were some lysimeters that wouldn't give a sample at certain periods of time. So rather than doing statistical analyses on the individual sample collection dates where we had missing data, we combined several sequential sampling events. So about 2 to 4 sampling events were aggregated together, and that sampling event was analyzed and compared to the other treatments on that sampling event. And these were differentiated mainly according to weather regime. So samples collected when it was really cold in the winter were analyzed differently than the samples when it was warmer in the spring. And also heavy rain events were used to differentiate the sample periods. So in order to get an estimate of the cumulative losses of nitrate, we had to calculate the volume of water that leached through the soil. The nitrate losses are equal to the nitrate concentrations during that given sample period times the daily volume of water that drained through the soil if there was enough precipitation to cause drainage. And drainage was calculated using the Food and Agriculture Organization method published by Allen. And this is a rearranged equation, and that just says that the daily amount of water that's lost via deep percolation is equal to the daily soil moisture deficit relative to field capacity. And that's minus the surface runoff. And that was calculated using the standard NRCS curve number method. The curve numbers were determined based on soil hydrologic properties from each field, and that was assessed with Web Soil Survey. When we worked on the Eastern Shore and we worked on large fields where there was multiple soil hydrologic groups within each field. We did a weighted curve number by multiplying the percent of total number of acres within each category. We got one weighted curve number for the whole field. Daily evapotranspiration was calculated using the Penman-Monteith equation, and that was parameterized with weather stations that were on site at the Central Maryland Research and Education Center in Beltsville, or on the Eastern Shore where we didn't have weather stations on site. An interpolation was performed between multiple nearby weather stations, and that was done using the Oregon State PRISM system. So as part of these calculations, we got one drainage value for each field, and we assumed that there were no differences in the volume of water drained due to the cover crop treatment. So even though that it's possible the cover crops could have increased evapotranspiration, which would have affected the drainage volume, we assumed that there was no difference. We didn't want to kind of build this difference into our estimates. So this is probably a conservative estimate of the total cover crop effect on nitrate leached. And any differences that we calculated on the same field are solely due to nitrate concentrations measured in the lysimeters, because each field only has one drainage volume calculated. Moving on to experiment A. This was performed 1 year, and it was performed on 2 fields on a downer loamy sand, as well as a russet silt loam. And the cover crops were seeded via drill at what is an ideal cover planting time in Maryland, August 26th. This is pretty early. And this was done into an open field where soybeans were mowed down. And we did this just to represent how these cover crops could affect nitrate leaching when they're planted at an ideal time, and comparing of one species to a mix. So our treatments were a forage radish, a cereal rye, a 3-species mix, and a no cover control. And the seeding rates used are in parentheses. And we installed lysimeters at 50, 75, and a 100 centimeter depth. That was done so that we could determine nitrate concentrations at this depth to see how it changes moving deeper through the soil profile over time. And greenhouse gas chambers were also installed. Biomass was sampled at late November and in late March. Moving on to some results. There was no effect of soil texture on fall cover crop biomass, and there was no interaction between soil texture and cover crop treatment, but there was a difference between the treatments. Highest cover crop biomass recorded was in the radish treatment, followed by the 3-species mix and the rye. And because we planted at an ideal planting time for the radishes, they grew very large. This is a pretty high biomass recorded. And the 3-species mix had about 70% of its total biomass made up of the radish. And comparing at a glance the fall cover crop biomass to the spring cover crop biomass, there wasn't a significant increase in rye biomass in between the fall and the spring. So that means the rye didn't do much growing. It didn't perform much action in between late December, early December, and the end of March. This scatter plot shows the nitrate concentrations, and that's underneath the no cover crop control treatments, separated between the sandy and the silty fields. Each dot is 1 lysimeter sample over time between November and March, and they're separated 50, 75, and 100 centimeters. So consistently, the pattern that we saw under the no cover treatment is that the nitrate concentrations started high and they declined during the winter at all 3 depths sampled. And the decrease at 50 centimeters happened sooner than at the other depths. And that's happening because the nitrate is washing down and it's not necessarily being replenished by surface nitrate as fast as it's leaching away. So 50 centimeters declines first, followed by 75 centimeters, and then 100 centimeters. This graph shows the average soil pore water nitrate concentrations, and this is the 75 and 100 centimeter depth average together, separated by the month when the sample was collected and cover crop planting treatment. So looking at the total annual average, there was no difference between the radish, the mix, and the rye. And as well as looking at December, January, February, and March. However, in November, the mixed treatment was more effective at decreasing nitrate concentrations than the rye was. And this was an unusual year in that it never got cold enough for the radishes to winter kill, but several times the weather got cold enough to damage the radishes, which should have released some nitrate, but we didn't pick up this release of nitrate in our lysimeter samples. So throughout the time our lysimeters were out, it didn't leach deep enough for us to pick that up. And consistently, all three of these cover crop treatments, regardless of what time we're sampling, decreased nitrate concentrations relative to the no cover controls. And they did this a lot more in the fall when concentrations are highest. And that's when they've been most active, was earlier on. So this scatter plot shows the cover crop biomass at both sampling times. So this is the fall sampling event and the spring, and that compares to the mean soil pore water nitrate concentrations. So each dot that you see is from one lysimeter and the plot average where the lysimeter was taken. And the fall biomass was more important at driving this relationship than the spring biomass was. And that's because most of the action that occurs is dependent on the fall biomass. They're growing earlier than this time when we started sampling, and they're not doing much in between November and March. So moving on to experiment B, now that we've established that using a mix can be effective at reducing the nitrate leached, we wanted to determine when would be a good time to plant it. So for this experiment, we had a 3-species mix, and this was performed on 4 site years. So that's 2 years, and each year we had a loamy sand and a silt loam field that were used. These fields previously were in wheat, and that was harvested in July. So while most farmers aren't just going to have an open field to use, we just wanted to do some mechanistic work and see the planting time that was important for the cover crops. And in order to ensure that we would be able to actually measure the nitrate in the soil, manure was applied at a rate of 67 kilograms of plant available nitrogen per hectare, 1 day before the first cover crop was planted in the fall. So on our main plots, this was a grass, clover, and radish mix with the seeding rates in parentheses. And this mix was drilled into the soil at 4 different dates in the fall. And each of these planting dates were about 2 weeks apart, starting in August and running through October. And this was compared to a no cover control. This image was taken from this experiment, and it was from May 7th. This just shows the importance of planting early on what shows up in the spring. So even though it's the same planting mix in both of these images, the difference is when they were planted in the fall. By planting earlier, there's more legume in the mix, which has positive dynamics in that it fixes nitrogen and adds nitrogen to the soil surface at a time when the farmer needs that. So this could potentially replace some fertilizer. Whereas when the mix is planted later, there's a lot more grass, which can mobilize some nitrogen and therefore more fertilizer needs to be applied at cash crop planting time. And on these plots, we measured cover crop biomass and nitrogen uptake both in the fall and the spring. And each plot had one lysimeter at 1 meter depth. Moving on to some results. This shows the relationship between the growing degree days available in between cover crop planting and December 1st is a cutoff, and the cover crop biomass that was harvested in the fall. So each of these lines includes 2 site years. The 2 years we worked on the sandy soils are in the blue line. The 2 years in the silty soils are on the orange line. And growing degree days accounted for about 75% of all variability in fall biomass. And the biggest beneficiary of planting early was the radish. By planting in August, the radish made up about 85 to 90% of the total fall biomass, whereas when the radish was planted in October, it only made up about 25 to 30% of all biomass that was harvested in the fall. And this difference was due to just planting date. There were no differences due to soil texture effects. So moving on to nitrate leached, each of these bars shows the cumulative mass of nitrate leached according to the previously presented calculations. And these are separated by site year, and each bar is from one planting tree. There was a difference in cumulative mass of nitrate leached due to a soil and year interaction, but that was an interaction with planting treatment. So therefore, each of these fields were analyzed separately to determine how effective the planting treatment was on each site year. Consistently, cover crops reduced the mass of nitrate that was leached. And the cover crops that were planted in August and September, so that's the green and the purple bars, those were the most effective in reducing the mass of nitrate leached. And on 3 out of 4 of these site years, there wasn't a significant difference between the no cover control treatment and the cover crop that was planted in October. This scatter plot just shows the relationship between total fall cover crop biomass and the cumulative mass of nitrate that was leached. And the 2 years are separated, 2016's the blue and 2017's on the orange. The relationship was stronger in 2016, and that's largely due to having larger cover crops. We planted a little bit earlier in 2016. And as you can see, there's a lot more variability at low cover crop biomasses. So when cover crops grow large, they consistently reduce nitrate leached, whereas at low biomass, it's kind of hit and miss how effective they could be. Okay, so now that we've established that planting a cover crop earlier is a good way to reduce the mass of nitrate leached, we wanted to move on to something a little bit more practical. So in this experiment, experiment C, we interseeded a 3-species mix into a standing soybean, and this is just a timeline of some field operations that took place. In Maryland, soybean senescence begins in late August, and that runs through for a little bit. Once the leaves start yellowing, that's when we planned our first interseeded treatment, and that was a little bit different, the timing between the 2 years. The soybean leaves begin to drop around mid-September, so that was the timing when we planned our second interseeded treatment into a standing soybean. And after the leaves drop, the soybeans still have to dry out a bit. They can't be harvested yet. So it's still a few more weeks before soybeans could be harvested. So these are just the 4 dates for this experiment when soybeans were harvested. The earliest one is just because that was a dry year and a dry soil. And then the drill treatment has to occur after the soybeans have been harvested. So that's typically a few days, up to a week later in this experiment, depending on how much it rained. And the interseeding was performed using this machine, which broadcasts the seeds into a standing soybean. And if they successfully emerge after a few weeks, then in September, they'll look like this. So this image is just a ground view of this experiment showing two adjacent treatments, the one that was drilled after harvest, October 24th, versus an interseeding, September 22nd. And this image is taken November 7th. We performed this experiment on 2 years, each year on a loamy sand and a silt loam, and using a rye, clover, and radish mix. And we installed lysimeters in each plot at a 100 centimeter depth. We installed greenhouse gas chambers in the drilled treatment, in our most successful interseeded treatment, and in the no cover treatment in 2018. And biomass was harvested in the fall and in the spring. So moving on to some results of cover crop biomass. In 2017, the earliest interseeded treatment was the most successful. And this is shown most in the radish, where it increased the biomass the most on both soil textures. And in 2018, we interseeded a little bit too early, August 24th. And so some of the cover crop seeds, they emerged and then they died. So in 2018, the treatment where we interseeded at leaf drop was the most successful, and that increased cover crop biomass relative to drilling after harvest. Okay, so these graphs show the results of the calculated cumulative mass of nitrate that was leached. And this is throughout the sampling period, beginning with the date when our first lysimeters samples were taken and ending when the lysimeters were taken out of the field. This is just the two sandy sites where we did this experiment in 2017 and in 2018. And we had more nitrate that was leached in 2017 and on our sandy soils than on the silty ones. So as these lines go up, that shows that was a calculated rain event that was heavy enough to cause some subsequent drainage. And the only difference between these lines on the same graph is due to our actually measured nitrate concentrations. In 2018, the cover crop that was drilled after harvest wasn't any different from having no cover crop at all. So that one was planted October 17th. And in 2017, the earliest planted cover crop was most effective at reducing the mass of nitrate leached. In 2018, both of the early ones were more effective than the no cover. So the cover crops effectively reduced the mass of nitrate leached on the sandy soils in this experiment. There wasn't much nitrate leached from the silty ones, but there was some signal from nitrous oxide emissions. There was more nitrous oxide that was produced under the silt loam versus under the loamy sand. And each one of these peaks shows one of our sampling events targeting when we believed that would be a good time to sample due to high nitrate availability and denitrification events. So this includes the time when the radish winter killed. Over the winter, these were soil freeze-thaw events. And over the summer, when liquid nitrogen fertilizer was applied to the following standing corn crop. And so emissions were greater during the summer following fertilization than they were during the cover crop growing season. And there was no difference in cumulative nitrous oxide emissions or nitrous oxide emissions at any sampling date between these cover crop treatments. So on these fields, cover crops did not affect nitrous oxide emissions. So moving on to experiment D, now that we have some results that show interseeding can be successful at reducing nitrate leaching, we wanted to try this at a large scale and on realistic operations rather than just under our controlled conditions at Beltsville. So we partnered with several farmers on the Eastern Shore, and we did this work over 3 years. Over those 3 years, we had 18 fields. Each field was set up as one replicate with all the treatments applied. Treatments were randomly assigned to a location in the field strip. And in 2018 and 2019, we had a no cover control strip between them. This image just shows a seeding map that we gave to one of our farmers, and it's superimposed with the underlying soil map. And measurements were made within each treatment along a transect. That's where the arrows are. That shows where we installed and sampled the lysimeters and where we sampled the biomass. And the flown treatment was aerially interseeded into a standing cash crop, either corn or soybeans, via an airplane. And the drilled treatment was drilled after the cash crop had been harvested in the fall. We measured cover crop biomass, nitrate leaching, and uptake. And this image just shows a ground view of the previous image. It's the same field. And this was taken a month after the drilled treatment was done. And this just shows the head start that the aerial seeded treatment has. Moving on to cover crop biomass results in the fall. In 2017, we had 7 fields. In 2018, we had 6 fields. And in 2019, we had 5 fields where we worked. So flying cover crop seeds early consistently produce the largest cover crops. And in 2019, we had one major exception to the rule. There was one field where we tried this experiment where the flown cover crop failed. Just visiting the field, it was really patchy. We would see a large radish a few meters away from the next one. So in order to account for the variability, we averaged a 0 into this flown from that field. And there was variability in all these treatments, and that differed between each year. But the brassica biomass was consistently high when they were planted early. These two scatter plots show the importance of planting early, and each dot is the average biomass from one field. So it's separated between the flown treatment in blue and the drilled treatment in orange. And in 2017, the flown was more variable, and that's largely due to rainfall. In 2017, there was 4.3 centimeters of rainfall, and in 2018, there was 19.5 centimeters of rainfall. So the aerial seeding needs rain in order to germinate. And not only that, but dry conditions are more favorable to drilling because when the field is dry, the farmer can harvest earlier and get the drilled crop in the ground earlier. And when it rains, that causes the flown seeds to emerge well. So when there's a lack of rainfall, there's more variability, whereas when there's plenty of rain, the amount of growing degree days available limited the growth. This graph shows the cumulative mass of nitrate that was leached by cover crop treatment. So overall, across 3 years, we had 18 site years, and this is made up of 46 treatment observations. So across these 3 years, we had 16 observations from fields that were sandy loams and 30 observations from silt loams. There was no difference in nitrate leached between the soil types. We also had 21 observations where we worked following a fall soybean crop and 25 observations where we worked following a fall corn crop. And again, that was not significant. But the cover crop treatment was significant. It did affect the cumulative mass of nitrate leached. And there was also some interaction between cover crop planting treatment and the year of the experiment. So overall, The cumulative massive nitrate leach across all 18 site years, the flown was equal to the drilled, but both had less nitrate leach than the control with no cover treatments. On 8 of our site years, the flown was more effective than the drilled. The drill was more effective on 6. So looking at the differences between the years, 2017, we don't have a no cover treatment. So just comparing drilled to flown, these were equally as effective to each other. 2018, during a rainy year, the flown was much more effective than drilling a cover crop, and the drilled treatment wasn't different from a no cover control in 2018. And 2019 was another dry year, and statistically, there was no difference between drilling and flying cover crop in terms of nitrate leach this year, but both had less nitrate leach than the control treatment. And on the Eastern Shore, we only had cash crop yields for 1 year, and that was 2018 to 2019, and the cover crop treatments did not affect the following corn crop yield. So this is a graph that I've shown before, but I just wanted to show that even at a large scale, this relationship still holds with each point here is one experimental unit from one field, and the fall cover crop biomass had an effect on the cumulative mass of nitrate leached, even though there's lots of variability. The biomass accounted for about 41% of that. We expect that once we have our nitrogen tissue data, then we might be able to account for more of this variability. That image will be coming soon. Across these 3 years, we found the weather was extremely important. So the precipitation in September, that's cumulative for the whole month. And to get these data, I use the Oregon State Prism system to take weather data at 3 locations that were central to all of our fields and average them together for each year, 2017, 2018, 2019. And in 2018, that was the year that favored flying due to heavy rainfall. 2019 was the year that favored drilling due to extremely low rainfall. And in 2017, the cumulative mass of nitrate leached was equal between the 2 treatments. And this was also a dry year where there was about 40% of the normal amount of rain. This is the mean September rainfall between 1981 and 2020. And just as a quick and dirty basis of comparison, the MAX model estimated that cover crops could reduce nitrogen loading by 8 kilograms per hectare. And this isn't directly applicable to the max numbers because this isn't loading. This is just leaching losses that are prevented. Obviously, other dynamics will occur after the nitrogen leaves the field. But the drilling treatment prevented leaching losses, averaging these 2 years of about 27 kilograms of nitrate nitrogen relative to the control. And the flown prevented leaching losses of about 32.5 kilograms nitrate nitrogen relative to control. So wrapping up with some conclusions, the fall cover crop biomass was more important than the spring cover crop biomass to reduce the nitrate leaching. With greater fall biomass comes less nitrate leaching, and that's because the early action is important. The cover crops aren't growing very much over the winter, so in the fall, they've already done their job. And when using the same planting method, planting earlier reduced the mass of nitrate leached. The cover crop effect on nitrous oxide emissions and winter nitrous oxide emissions that we measured were negligible on these fields when compared to the peak following fertilization. Across all of our experiments, we only had 1 site year where the cover crops affected the following cash crop year, and that was just due to it being a really dry summer on a sandy soil. There was no effect of a cover crop on the following cash crop yield on any of our other experiments. So while interseeding success was variable, on average, the earlier start of planting a cover crop seed in the fall was more important than the effect of having a patchy stand when it comes to nitrate leaching. In our wet year, interseeding was more effective than drilling at reducing nitrate leached. In the dry year, both treatments performed similarly. So obviously I couldn't have done this work alone. If you're a student with your name on the screen, you did this work with me for 2 years. There were 23 others I couldn't list. I'd like to thank all of you. I'd like to thank our Eastern Shore collaborators and my committee, funding sources, and the staff at CMREC. And with that, I am happy to take any questions.
Thanks a lot, Nathan. Really interesting talk. For those of you that are guests, we typically do questions by putting things in the chat box. And so let me do a couple reminders quickly as people are writing, or if you just want to turn on your mic and ask a question, that's okay. We have a lot of people on today. Just a reminder, if you wanted the CEU credits, please go ahead and send a chat to Emily Lucas, who can take care of that. And if you would be so kind as to fill out the evaluation form that Dr. Ravenhorst has put a link in the chat box to give Nathan some feedback on today's talk. So with that, are there any questions? Let's see. So I guess I had a question, Nathan. So, if you're put your future extension agent hat on, and so if a farmer was interested in doing the aerial seeding, what kinds of suggestions would you give given the weather variability that you saw? I mean, and do you think there is ways to improve that technology?
Well, with the weather variability, I do want to establish that, we were comparing on the Eastern Shore an extremely wet year to 2 abnormally dry years.
Right.
One of the years that was pretty dry in 2017, they still performed similarly that year. We didn't have failed stands that we observed in 2017. It was only in 2019 where it was actually the 40 years we looked at, it was the ranked 38th in terms of total rainfall. So one of the driest years we ever had, there was a failed stand. So I think that during the dry years, it's likely that they'll probably perform similarly. You know, there's still a risk of failure, but during a wet year, the difference isn't even close.
Mm-hmm. Do you think that those results can be extended beyond just the Eastern Shore into other types of soils if we're talking, in the Piedmont as well? Or is that unique to the sandier soils there?
Well, throughout this research, I didn't find an effect due to comparing sandy loams versus silt loams in terms of cover crop establishment. So while I'm not as familiar with the soil types in the Piedmont region, I could at least say that in terms of the soil types that we looked at, there wasn't an effect on cover crop interseeding success.
Let's see. So there was a question from the audience about, are there other entities that are doing or have done this kind of field research?
In terms of entities, I know that there are people at the USDA that I've talked to that do some interseeding cover crop work, and Steve Mirsky is one of the first people who comes to mind. He actually had one of his technicians set up an experiment on the same fields we used on the Eastern Shore for a couple of years.
Oh, okay. Yeah. Are they doing some of that aerial seeding as well? I think that's fairly unique, right? I mean, in terms of, that hasn't really been done at that scale before, has it?
Well, I know that interseeding via an airplane is a common practice on the Eastern Shore. I don't know if Steve is testing that specifically.
Okay. So there's a couple other questions. What do you think are the best ways to get more farmers to adopt cover cropping practices? Thinking many may be hesitant to alter those processes that they've been doing.
Well, certainly the cover cropping program that we have in Maryland has been very successful at incentivizing cover cropping. I mean, we have the highest percentage of cover cropped acres in the nation, and the incentive structure that they set up for aerial seeding has also, I mean, they've been pretty dynamic with incentivizing earlier planting. So I think that their program is doing a good job. They just, need to move a few deadlines as the research becomes available, if the data supports it.
Yeah. Do you know if other states, I mean, I know Maryland's been really a leader in that. Do you know if other states are starting to adopt sort of similar practices?
In terms of paying farmers
Paying
to
for
cover crops,
cover crops.
that I do not know,
Okay.
but it's certainly something I could get back to you on.
Yeah, I was just curious. It's something, I know Maryland's done it for quite some years now, and it seems like we've got an accumulation of data that it's helpful to do that. But I don't know. So let's see. Other questions. So what about the economic considerations for farmers choosing to do cover cropping or not? So I guess that gets at that a little bit at that question of if you're not incentivizing it like you are in Maryland, if you're gonna start a farm in, well, Dietrich asked the question. He's out in West Virginia now. So how about that in terms of considerations?
Okay, so the cover crops can also provide a benefit in that at least the legumes can replace some nitrogen fertilizer, which could save the farmers some money. They have some soil health benefits. I didn't explicitly do that type of work, but I know that the research exists.
Okay. Yeah. And let's see, we'll ask just a couple more here as we get to the hour. The first is, were you surprised that there was no effect of cover crops on the next cash crop? It's a little disappointing that there wasn't a yield bump.
Yeah, so my understanding is that there's some research that exists that shows that at least planting a grass can have a yield drag on the following cash crop. So my instinct also was to be disappointed by that, but after talking to Ray, he let me know that, hey, it's not a bad thing that they didn't have an effect. It's common that a cover crop might reduce the following cash crop yield, and that's just if you use a grass that might drag the yield of the following cash crop.
Is that a competition for nutrients, I guess? Is that the drag?
Yeah, because they're decomposed slowly.
Gotcha. Okay. And let's.
Can I just add to that?
Yep.
Just to know, these were all single year effects of cover crops, and all of the fields had a history, because it's in Maryland, of using cover crops. So you're taking fields that normally have cover crops, and all you did was leave the cover crop off for a year.
Right.
Okay, so the long-term effect of cover crops probably does improve crop yields, but these studies weren't set up to measure that.
Let's see. So someone asked about the seeding rates of your cover crop species.
Yeah, so at Beltsville, the mix that we used was what was called the Charlotte mix. And let's see, I have those pulled up on a slide here. And let me go back to sharing my screen. So for the mix that we used, it was 84 kilograms per hectare of rye, 6.7 of radish, and 13.5 of crimson clover for the mix. And the cereal rye amount was designed to comply with the cover crop program guidelines. And the mix, the seeding rates were used consistently at Beltsville. On the Eastern Shore, we asked our farmers to use grass, a legume, and a brassica in their mix, but we also told them that they could use the seeding rates they felt comfortable with, whatever their typical practice was. So that one varied over time.
And we'll ask one more question, and that question was concerning the manure application and how that was applied.
Yeah, so the manure was spread onto the surface of the field. So these were just open fields that previously there was wheat harvested. And so at the Beltsville facility, he just spread dairy liquid manure onto the soil surface.
All right, well, we are at our time. I know there's a few questions that were left in chat. I've actually saved the chat and I will email it to Nathan so that he can see those questions, and possibly we can, this talk is been recorded, and that recording will appear also on the ENST webpage. And so there's another way to reach back out to Nathan. I think we usually put emails down if you're interested in things. So with that, I think we will finish up unless there's any final reminders. I see Emily there. Are you good, Emily? Okay, so we're good. And thanks again, Nathan, for a really interesting talk.
Thank you for attending, everyone.